Okay, we're going to run through the notes for Chapter 14, the human genome, and today we're probably just going to cover the first little bit of that, working up to pedigrees, and maybe getting into some blood typing as well. So the first thing, this is actually from Section 3, but I wanted to make note of it right away. The Human Genome Project was a project that scientists kind of underwent in order to figure out and map the entire sequence of DNA inside each of your cells. So they started this in 1990, they finished in 2003. And so while they were doing this, they just found the sequence of all 46 chromosomes. And obviously, with it taking 13 years, they found a lot of different information on our chromosomes. So they mapped where every specific trait that you get and where that trait is located in the chromosomes. About 6 billion nucleotides were sequenced, 23,000 genes in each of those genes, then creating either a protein inside your body or the specific traits that you see with your nose shape, hair color, eye color, those types of things. So if we look at these two karyotypes, and a karyotype is just a sequence of chromosomes showing all of the different chromosomes that an individual has. We can see two key differences between each of these. They obviously each have two sets of chromosomes. The one on the left has 23 pairs of chromosomes. You can see the X and the Y down here at the bottom. So this is obviously a male individual. And the one on the right has a little bit of variation compared to that. So if we see, we now have 48 chromosomes, or 48 total chromosomes, and 24 pairs. And so what we have here is still an X and an X, so this is a female individual, but this is actually the genetic information for a chimpanzee. And so the chromosomes themselves are pretty similar, and we may eventually cover a little bit more on what the key differences are between chimpanzee DNA and human DNA. Okay, so quick review on human chromosomes. First thing is that humans have 46 total. Okay, remember, we have 23 pairs are in each of your cells. Pair number 23 is the sex chromosomes. If you can look back to the previous one, our pair number 23 down here was our X and our Y. So that's what determines the sex of the individual. And two X chromosomes create a female. One X and one Y is the male individual. So we can see the difference between those two. All right, we got a couple different types of chromosomes. First of which, meaning the X and the Y, are the sex chromosomes. That's the last or the 23rd pair. But the other chromosomes that are not involved in determining the sex, we call autosomal chromosomes. And so this is the other 44 chromosomes. You have 22 pairs that are not the X and the Y chromosome. So autosomal chromosomes just mean everything else that's not a sex chromosome. So the proper way that we write the sequence for an individual is showing that there are 46 total chromosomes, and so this is with humans, and then XX represents that is a female. With males, 46, again, saying the same number of chromosomes, but now an XY, and that tells us that it is a male individual with 46 chromosomes. So eventually we're going to talk about some disorders that change the number of chromosomes that an individual has, and so that's why it's important to have the 46, then the X and the Y to show that it's male 46. All right, the way that offspring are actually created, okay, we know during meiosis, the 4 daughter cells are created, the sperm or the egg. And so this shows us how we determine whether or not an offspring is going to be male or female. So the female only has the ability to pass on an X chromosome and an X chromosome. So all these will at least have one X, which is why everybody has an X chromosome. Males have either an X or a Y. So with our Punnett square, we can see that this is how we get a 50-50 ratio of male to females when individuals are born. So there we get our 23X plus 23X for this individual gives us 46XX, and then 23X plus 23, then a Y chromosome gives us our 46XY for the male gametes. All right, we covered pedigrees in class, but I'm going to just review this just in case anyone was absent. So pedigree, just a chart that we use to show how specific individuals are passing on a trait. And so a lot of times dogs use this, or people use this to compare the relationship with dogs and their ancestry. pedigree or the more pure line that it is, that generally indicates that the dog is going to be more expensive or you have to pay more in order to get that dog. So we use this to study inherited traits. We can look at anything from hair color up to disorders like hemophilia or some other ones that we'll eventually talk about, like PKU and some other ones. Pedigrees are often used in genetic counseling. So if you have spouses that are looking to have children or planning on having children, but they may have a chance of passing on some type of disorder like hemophilia, they can go into a genetic counselor, they can pull up and chart the pedigree of each of those individuals and then figure out what the chances are of their offspring having hemophilia or whatever other disorder.
Good morning.
And we're back with the bells now ringing. So last thing with those pedigree cards, we can look at both dominant and recessive traits. Those are probably the easiest to interpret using a pedigree, but we can look at some other things that are a little bit more complex, like a codominant disorder that we may see, or that we may see throughout human inheritance. All right, so how to read a pedigree, we're going to look at a couple of different things on this chart. First thing to notice is that we will see squares and circles. And if I flip to the next slide, this is an actual pedigree. You can see squares and circles, and that's about the only different shape that you see throughout that, because square is representing a male, and so filled in means that that male has either the disorder or the trait that we are looking at. A blank square just means that it's a male that doesn't have that particular trait. Okay, so after that interruption, Logan's still hanging out here. Circle represents a female. So a blank circle, female that does not have the particular trait, filled in does have that trait. So it does have the disorder, does have whatever trait it is that we're looking at. As we move down a little bit lower, okay, a circle and a square connected by a line represents mating or marriage. And then two lines mean basically a divorce. They're no longer together, but they possibly produce offspring at one point. When we have a vertical line coming off of that marriage, that represents the offspring. And so in this case, a square offspring represents the son, or a son. In this case, the parents had 4 children. All right, I forgot where we're at, because we had a couple people in a row. So square represents male, circle, a female. This particular couple had 4 offspring, the oldest of which always goes on the left-hand side. So oldest was a male, then a female. I'm not talking to you, Derek. I'm making my notes. And then finally they had a set of twins, 1 female, 1 male. So non-identical twins come off the line at one point. If they were identical twins, the line would come down vertically first and then diverge off into the other 2 individuals. Okay, so we're going to cover more on pedigrees tomorrow during class, although I'm not going to go over these examples right now. And we'll cover this tomorrow as well. Okay, so we're going to go through a variety of examples on pedigrees, but now we're going to cover just a little bit on genes and the environment in order for us to be ready for class tomorrow. So a lot of environmental factors can play a role in what traits are going to be represented by an organism. So it's not just what an individual is going to get from its parents. That's the biggest portion, but also things like nutrition and exercise. We're going to have play a factor in what that organism is going to look like. Obesity, for example, is caused by that. So if we look at height, for instance, in the last about 200 years, the average increase for humans have increased about 10 centimeters, which equates to almost 4 or 5 inches. And so that is based mostly on the nutrition, the food that we're eating, and that's going to be changing what type of overall phenotype is created, but it's not changing the genetic information at all. It's just because of the food that we're eating being a little bit healthier or a little bit different as far as what materials are put into that food. It changes how the phenotype ends up being represented. And let's bring the last slide on this video so we can keep it to 10 minutes. we're going to talk a lot about blood typing. Blood typing is an example of multiple alleles. And so with these multiple alleles, if you remember back from our rabbit color square that we were talking with, where there are various different combinations that make either the full color or the chinchilla color or the Himalayan, blood typing in humans is very similar to how that worked with the chinchilla rabbits or the full-colored rabbits. With blood typing, we actually have two different parts to this, the first of which is the Rh group. And all this is either a positive or a negative that gets tagged on to whatever blood type that you have. And so it's called an Rh blood group because it was first discovered in a species of monkey called the rhesus monkey. And so they give it the Rh factor representing that that's where it originally came from. All right, so this is where we're going to stop with the notes for this video. We'll see you tomorrow.